Hi, my name is Allison Beal, and today we're going to be talking about the Phylum Phoronida. First, we're going to go ahead and talk about the phylogeny of these animals. And if we look at the diagram, we can see that they are located underneath Lophotrochozoa, and more specifically so, Trochozoa located here. And if we follow the diagram all the way back, we can see that they are related to Bilateria, which will prove to be important when we are talking about their development further on. We're going to go ahead and move on to ecology. These animals are marine benthic tube dwellers, and they are mostly euthermal and euryhaline, meaning that they are able to withstand a wide range of salinity and temperature ranges. And because of that, they tend to have a worldwide distribution, and they live more specifically on substrates and in benthic communities. However, if currents are strong in these areas, their distribution tends to be limited, especially so if these strong currents are occurring in or below the intertidal zone, which is where they will be most impacted by those currents. Now we're going to discuss the basic body plan. They range from 1 to 25 centimeters in length, so they're fairly small, and they exhibit little regional specialization other than their lophophore and their end bulb. Their lophophore is located here, which is the large feathery portion that is sticking out of the tube, and the end bulb is located down at the base of their tube, which houses the testes, ovary, and intestine of these animals. They actually have a very interesting circulatory system, because rather than having solely a closed or open system, they actually have one that is both closed and open. Their circulatory system consists of two large vessels that run along the length of the phoronid internally. One is the efferent vessel and the other is the afferent vessel. And the blood flows down the efferent vessel and ends up in the end bulb. This is where the system stops becoming a closed circulatory system and rather becomes an open circulatory system. When the blood reaches the end bulb, it has to move through the spaces of the organs to reach the afferent vessel. In the period between the efferent and the afferent vessel, it is an open circulatory system. Then once the blood enters back into the afferent vessel, it once again becomes closed. The blood moving through the circulatory system is caused by contractions of blood vessel walls, and we are now going to talk about feeding and digestion. And these animals are considered to be suspension feeders. So they use their tentacles, which stick up out of their tubes on the lophophore, to catch food and bring it into their mouths. They have a U-shaped gut, and as larvae, they have an oral hood that is encircled by moving cilia. So the cilia are what move the water over the hood towards the mouth. And in doing that, they are drawing in particles of algae and other organic matter. And the oral hood will only open after it has made contact with one of those organic particles. And then once the oral hood opens, which is due to muscle contractions, the opening of the hood produces a suction, which pulls in the water that has those organic particles within it. For their nervous system, they lack a distinct cerebral ganglion, which is most likely caused by their sedentary lifestyle. They don't really go anywhere. And instead, the central nervous system is a concentration of neuronal cell bodies that is located between the mouth and anus, which, because it is a U-shaped gut, is located right next to each other. As you can see on this diagram here, the anus and mouth are next to each other, and their central nervous system would be located right between. One more, all right. And throughout their body, they have a layer of nerve fibers that are located between the epidermis and circular muscle layers. And from this layer of nerve fibers, they have sensory neurons that project outwards through the body surface. And those sensory neurons are the only receptor structures that these animals have. As for reproduction, they are both gonochoric and hermaphroditic. They typically have internal fertilization, however, external fertilization can occur. For internal fertilization, the spermatophores are transferred to the metanephridial opening of the females, and at that location, these spermatophores will release the sperm, which travel down the metanephridial funnels, where they will land near the ovary, which again is in the end bulb of the animal. For external fertilization, the spermatophores land on the tentacles of the lophophore, where they release the sperm, and the sperm then travels through the tenticular wall, through the mesocele and metacele, into the trunk coelom, where the ovary is located. So again, it's moving back down into the end bulb. And they are also able to exhibit asexual reproduction through fission or budding. For development, they're able to exhibit radial, spiral, or indeterminate cleavage, and the development of their cleavage pattern depends on the size of the egg as well as whether the embryo is brooded. For broadcast spawned ova, they have a very small yolk, and because of that, they quickly turn into planktonic actinotrope larvae. And for species that have nidamental glands, they produce eggs that are moderately rich in yolk, so they don't turn into the larvae as fast, and instead are brooded until they reach their actinotrope stage. And this right here is an example of one of the actinotrope larvae. For their tube formation, they are secreted by the epidermal gland cells, which are the A and B cells, which are acetophilic and basophilic. In acetophilic A cells, they produce mucopolysaccharides, which make the thinner, compact peripheral layers, and the B basophilic cells produce sulfomucopolysaccharides, which make the central basophilic layer. And here you can see two different examples of the same phyla. Something interesting is that relatively recently, in the early 2000s, there was a giant actinotrope larvae that was found off the coast of the South China Sea. And so these larvae are typically between 0.7 and 0.9 millimeters. However, this larvae was 3.5 millimeters in length and 3.5 millimeters in width, with 52 thin tentacles that were up to 1.4 millimeter in length, which might not sound huge, but if you look at this diagram, which is to scale, this is the size of the larvae that they found, and this is the size of a typical actinotrope larvae. So it is quite large. And these scientists who discovered this larvae feel that it is most likely not a discovery of a giant adult animal that we haven't discovered yet, but rather it is most likely a disturbance in the larval development that resulted in the larvae not being able to transition from the larval stage into the adult stage. And ending on a fun fact, the Phoronids were actually named after Phoronus, who is also known as Lo in Greek mythology, who fell in love with Zeus and was turned into a white cow by Hera as punishment, which I believe the reason that they are called Phoronus is because of that metamorphosis that the larvae undergo to turn into the full-blown adult. All right, that is it. Thank you for your time.